Hi, everybody. Thank you very much for joining. We'll just give another minute or two to get a couple more people in, and then we'll get started. Thank you again, everybody, for joining today. Just a few more seconds. Okay, great. I think we can get started. So, hello, everyone, and welcome back to part two of the 2025 GFI Research Grant Program RGP webinar series. My name is Arin Naidu, and I'm the Senior SciTech Ecosystem Specialist at GFI APAC. I'll be your host once again for today's session. Before we begin, I'd like to take a moment to introduce Alt Protein Dialogues, the broader webinar series under which this session falls. Alt Protein Dialogues is a new webinar initiative co-organized by GFI APAC, GFI India, and GFIC. This webinar series is designed to connect and highlight alternative protein innovators across Asia Pacific, India, China, Australia, and New Zealand. We aim to foster regional partnerships and drive globally impactful innovation in the alt protein sector. This two-part grant call webinar series is our first event under Alt Protein Dialogues, and we hope to build on this momentum to bring more knowledge sharing opportunities across the region. So please do share this webinar series or with your peers as well who might be interested. In part one, which happened last week, we explored the GFI Research Grant Program, its objectives, focus areas for 2025, and the application process. Today, in part two, we'll actually have guidance from past grantees. We'll hear directly from past RGP recipients who will share their experience, insights, and advice for future applicants. Our goal today is to provide you with practical knowledge from researchers who have successfully navigated the application process and secured funding. This session will help you understand what makes a strong research proposal, how past grantees structured the applications, lessons learned from their experience, and how this funding supported their work and career. So how today's sessions will unfold, we'll hear from 3 past RGP grantees. We're very lucky to have so much involvement today, each speaking for about 15 minutes, covering the research topic, which they focused on their RGP, application insights, and their experience as a GFI-funded researcher. After their presentations, we'll open the floor to a 15-minute Q&A sessions where you can ask your questions directly to our speakers. We would like to focus the questions on the application process and their experience, rather than deep diving into any of their research, as much of their work could be confidential at this moment. Now, let's introduce our esteemed speakers for today. Our first speaker is Dr. Deepti, a senior project research scientist from the India Institute of Technology, Bombay, and Clarity Biosystems as well. So currently two postings, and then she's currently working on developing and optimizing low-cost, serum-free culture media for cultivated chicken meat using metabolic modeling and spent media analysis, along with Professor Pramod Wangikar. After Dr. Deepti's talk, we'll have Associate Professor Dr. Juan Du from the Singapore Institute of Technology. Prof. Juan Du received her PhD in food science from Purdue University, and then went on to have relevant industrial translational research after graduation at Nestle Process Technology Center in Bakersfield, California. There, her work was focused on ice cream innovation. Currently, Dr. Juan Du is working as an associate professor in food technology at Singapore Institute of Technology, and she's one of the editors-in-chief of journal Food Biomacromolecule. Juan is responsible for a number of government and industry-funded translational research projects, including a high-profile program utilizing automation to characterize protein techno-functional properties and identify protein ID. She received the Young Alumni Award and now serves as a visiting professor at the Purdue University Food Department. In addition to research, she also currently serves on the Singapore Food Standards Regulatory Standards Committee and co-chairs courses sponsored by the UN Sustainable Development Goals. She also is one of the PIs of the Bezos Center for Sustainable Protein in Singapore. Our final speaker will be Dr. Nazneen Sofio. Dr. Nazneen Sofio is a senior specialist at Singapore Institute of Food and Biotechnology Innovation, also known as A*STAR. Her current work focuses on providing producing alternative protein lipids using a variety of techniques like enzyme engineering, adaptive laboratory evolution, and metabolic engineering. Dr. Sofio has been awarded with multiple grants to pursue her work in the field. She has expertise in molecular biology and metabolic engineering of the lipid biosynthetic pathway and strives to be at the forefront of lipid biology research. Before we dive in, I just want to do a quick housekeeping note. You'll see the chat and Q&A buttons at the bottom of your screen. So please use the Q&A function to submit questions for our speakers. And when we get to the Q&A session, we'll kind of pass through those questions. You can also feel free to upvote which questions you think we should prioritize as the most relevant ones. This webinar will be recorded, and we will share the recording along with additional resources after the session. The first webinar, if you did miss that, was also recorded, so you'll have access to that as well. So with that, let's begin. Once again, just thank you to all of our speakers for being here today. We're really lucky to have all of you and very grateful. And yeah, let's get started. I'll now hand it over to our first speaker, Dr. Deepti, to share her insights. Thank you very much.
Thank you so much, Aryan. I hope I'm audible to all of you. And so I'll share my screen now. Yes, so I hope all of you can see the screen. So great. A very warm good evening to all of you. And this is Deepti, and I work as a scientist at IIT Bombay. We have a startup company called Clarity Bio, which specializes in metabolomics and metabolic modeling, as well as AI-based modeling. And our expertise is observed to be very complementary to people who work in the alt protein domain. I'm really excited to talk today to all the participants who want to really, apply for the funding. And I must really tell you that it's an awesome opportunity. So not taking a lot of technical jargons, I would just quickly go through what do we do as a part of GFI funded project, an overall overview of the project, followed by what do we exactly do, and then we'll discuss about why GFI and how did we apply and what are the tips, which I think will definitely be helpful in longer run. So we have so the project we have is we're trying to optimize a model-guided media optimization for a chicken cell culture. And I think it's imperative that cultivated chicken cells or other cultivated meat is being on the forefront of this research. And we find our collaborators who have expertise in the chicken muscle cell culture. We also have another collaborator, and we really acknowledge them for this, who can actually give us their expertise in finding plant homologs and the protein research. Our expertise is in the domain of metabolomics, and today I'll be talking very briefly that how did all these various, I would say, expertise really played a complementary role in this GFI funded project. We all know that the major driving force in taking the cultivated meat to the commercialization has been, it's always it's high cost. The cost has been always a factor which has kind of been a pain point. And we decided to approach that challenge head on. But how did we decide to do that? We decided not to go by a traditional DOE sort of approaches where we try different combinations of media or maybe feed recipes and find which one is suiting best for our chicken cell culture. We decided to go by a very targeted, very very model-guided approach. For this, we need to combine a lot of omics technology, and metabolomics was very handy to us. So we decided to grow chicken cell culture in serum-free, and our recipe of serum-containing and serum-free media, we decided to understand what is it that cells are secreting and cells are taking up in the media. Once we have more clarity on the media, we actually decided to build something we call genome scale metabolic model. This is, you can imagine it as it's a digital twin for the cell. Of course, we're not yet there, but we're trying to actually model the entire cellular metabolism in computational space. So every dot here is a metabolite, every reaction is actually an arrow. we're trying to have multiple mathematical equations. We are trying to solve them to find out how cells behave. And if we provide enough data of surroundings, if we provide enough data for what cell is taking in or what cell is giving out, can we understand the physiology? Can we get the insights of the cell and we understand what is it that cells need to survive in the environment so that we can feed exactly that. Once we have insights, we can actually try and replace the animal proteins or animal components with plant-based components. Now, this is more directed approach because we already have an insight from the simulations which we run in our day-to-day experiments. So basically, we are combining our metabolomics, feeding the data into a model to make it something what we call as dynamic flux balance analysis. So basically solving multiple mathematical equations at once to provide insights so that we can have an optimized media. So this is what we've been all doing for the GFI funded project, which initiated in 2024. And as I said, I'm really grateful to all our collaborators who have provided us with their knowledge base in this particular field, as we were the new applicants in this domain. We've never worked in the high protein domain before. So what were we exactly doing? We were working in the field of metabolomics. What is all about? We were trying to characterize small molecules which are less than 1500 Daltons, and our expertise has been in this domain for almost a decade now. What we use is we use LC and GCMS to actually find out as many dots from these metabolic network as much as possible. So we either run it in the scan mode, we actually try and find every dot, of course, every dot is not possible, but we try and find maximum dots. Or we focus or zoom in on one particular pathway and we try and find, quantitate some metabolites out of all the network which we have. We therefore have a potential to analyze more than 500 metabolites now. If you really want to understand what are the concentrations on which we are working, glucose is if it's the human blood, glucose is at maximum concentration. It's like some milligram per deciliter. As we go down, as we go to vitamins and steroids, they go in the nanomolar range. And we have a capacity, now LC and GCMS has capacity to understand even at this stage. So we actually have complete understanding of whatever sample comes to us. While doing this, as I mentioned, we use LC and GCMS. Data acquisition is absolutely easy. I mean, comparatively, I understand it's not easy at all, but the challenge is data analysis. LC and GCMS provides such a huge amount of data that it might take 6 months to 1 year to just analyze a data from single set of samples. We therefore understood this need and we have developed a proprietary software based on AI/ML base, which helps us not only do a routine quality check of the data, but also analyze the data, identifying a peak versus noise very carefully, and also to statistically analyze the data. So this is where our expertise has been. We work in the chemical engineering domain as well as bioprocess development domain for some time now. And our group also has an expertise in something we call genome scale metabolic modeling. So just for giving a perspective, we start with an annotated genome. We start making a draft metabolic network. Again, this is all done in computation. We try fitting the dots, but trust me, the genome is always draft. It's not yet well annotated most of the times. So you have lot of gaps. The network is not complete. And we have to put additional effort, rather iterative effort, to complete this particular genome scale model using multiple omics data, functional data, data from actual experiments, etc. Once we have a model, we try and acquire more experimental data at multiple time points, providing constraints to it, so that imagine it is solving multiple mathematical equations at once, which will help us in so we have an objective function. So we have a clear goal in front of us to improve biomass. And then we solve these equations, keeping that as our goal. And then we find a particular space where we understand in which environment cells are comfortable. Now, once we have that understanding, which is like a lot of insights, which help us in understanding what is required by cells. Can we feed that in? So it also helps in defining the recipes. Now, this model-based insights we use a lot on process intensifications or optimizations. And I think, as I was trying to mention just some time back, we are completely new to this field. We have been applying our expertise in fermentation, which is via spent media analysis. We've been using blood or even clinical origin samples. We were doing a lot of cells or bacterial analysis, also chemical origin samples we were trying to analyze. And now we are developing our expertise in an alt protein domain as well, using a GFI funded project. In our funded project, we already have developed a genome scale metabolic model, and we have a lot of progress which we've already done in terms of understanding what cells need and what cells are taking from animal source so that we can replace it using relevant plant source. So we have significant progress towards that direction. So I guess we're not going into a lot of technical details. This is where we come from. And trust me, GFI has been a great support for us. First of all, I have certain feedbacks from our entire group. It helped us in collaborating scientists from absolute complementary skill sets from various new domains. So as I mentioned, we had never worked in alt protein domain before, but we realized that we have skill set which can be borrowed and which can be used for this domain, and we can actually get results which are otherwise difficult to obtain. In traditional experimental, it will take a longer time. We probably can help to get it faster. The second and third point is also because we've been dealing with multiple other grant programs, we've been dealing with government funding programs and all, and I must agree that this has an easy application process. We had quicker review, clearer instructions, lesser bureaucracy, and when we are starting something new, something novel, I think what is most important is smooth fund dispersal. And when we have all of that in hand, we can actually, when we have support from GFI in these aspects, we can focus on our research and get things going. So I think that's been where GFI has really helped. And very few tips for the application for any grant program from GFI. I think what helped us is to have objectives in line with the problem statement. So GFI defined problem statement keeping not only the current research, but as well as future of given alt protein research. So we kept our objectives in line with the problem statement. We focused on societal impact of our solution and also defined a very clear strategy. It was not a very lengthy application, but a clear strategy to our solution. If you want to approach a problem using model, we just stated why model was important. We gave a literature support to proposed solution and then timeline for the project so that it helps us also in terms of tracking where we are going. We had collaborators, as I mentioned. Our expertise has been extremely complementary, and responsibility of collaborators were clearly jotted down while we wrote the project. And of course, when it comes to budget, very simple budget heads, no confusion there, and we just had predefined budget at the time when we submitted the grant. I think what helped us is shorter application comparatively, clearer application keeping to the point, literature surveys supporting the strategy. I think all that definitely helped us in writing grant program in a little more effective way. And what I must really tell you, I really want to thank here our lead scientist, who is Professor Pramod Wangekar. He's a professor at IIT Bombay, and now, as we said, he has a startup company which is incubated at IIT. Clarity Bio team. Of course, it's not possible only by one person's support. We have an entire team who are experts in AI/ML, mass spectrometry, and modeling who help us. And I mean, I just will not go without mentioning our collaborators. We have a company which works with us, MyoWorks, who help us in providing their expertise in chicken cell culture, and we have a lab from IIT Bombay, MLab, which helps us in giving perspective of plant homologs. How can we replace animal origin protein? And of course, GFI for their funding support, and this idea can actually come live just because of that support. So please feel free to contact us if required. And I think I'll stop here, but I must really appreciate the effort which GFI has been taking, and I'm so sorry. I think there's something which went wrong in between. Am I still audible?
You are audible, your video is off, but
Yes. Oh, I got that point, but I think that's some camera or there's something wrong with the video. But okay, never mind. I think I conveyed whatever I wanted to convey. I hope the slides were visible, right?
yes, that was wonderful. Thank you so much, Dr.
Yes.
Deep.
So thank you so much for all of you having us. We really feel great to represent Classify this project here, and I'm sure we're going to do amazing. All the participants, I wish best of luck to them.
Yes. Thank you very much, Dr. Deepti. I think it was really cool to see how you applied you and your collaborators' skill sets into like all proteins, and then how this grant call allowed you to enter this space. I think that was really exciting. Thank you for also highlighting how this space actually allowed like really strengthen the collaborations and that as a key point. I think at GFI, like RGP or not, like we always want to be supporting collaborations. So I think that's really exciting to hear. And then also like the detailed list of like tips was also, I think, really helpful for all of the audiences today. So thank you very much. And to the audience who like any questions, Q&A, please remember to start putting those in the Q&A box, and then we'll address that at the end. So yes, thank you again, Dr. Deepti. I think if there's no last words from yourself.
Well, I think all the best to the participants and all my colleagues who are talking to me today. I'm sure collaboration is extremely important, and at the same time, smooth administrative process for any funding is important. So thank you, GFI team, and all of you for that. Thank you so much for having us here today.
Thank you very much, Dr. Deepti. With that, I would like to pass over to our next speaker, which is Dr. Juan Du from SIT.
Thank you, Irene, and good evening or afternoon to all of you. It was my great pleasure, to join this event to share about my experience. I would like to grade my highest appreciation to Good Food Institute, specifically for this APAC region, because I have received significant support, not just for me, myself, and also the team who has been working and otherwise has significantly helped us to shape and make us who we are today, coming to our research direction. So I do have one slide to share about this project. Give me a moment. Share it. Sorry for the delay. So can you see my screen clearly? Yeah. All right. So this is our project, because I don't know what's a better way to show, you can see that in this project, we have involved quite a significant number of collaborators from research institute, as well as industry. The project is called Sea-to-Meat, which is optimizing seafood proteins to cultivate meat. Me, myself, before this project, I have no touch and experience when cultivating meat. My expertise really remains with plant-based food innovation, food processing, and this attribute. So luckily, because of some existing project that is ongoing, so Professor Angelo Lopata has visited our university, who has the expertise in seafood allergen, food, as well as proteomic profiling. So who came to us and understand that SIT has a food characterization as well as food processing facility available. So we were thinking how to link and together working on something that is contribute to the current food security for Singapore as well as beyond, because of the global growing population and the requirement of food, which is adding layer of stress and risk for the food security and the resource outside. So we were thinking, what shall we do? We started in the project and also the material, which is a problem, that is seaweed. Currently, most of the seaweed, especially the protein and other components, are discarded after the polysaccharide extraction. But you know that the protein is so critical, and their protein profile coming to essential amino acid and other components are quite critical for the human nutrition. We're thinking how to utilize this one to maximize the value adding. Therefore, we are considering, instead of considering doing intensive extraction, well, just utilizing it as a protein or some material which has already been done intensively in other projects that is showing there might be a chance of success, but the barrier of separating the protein, to make it intact, well, increasing the yield or maintain the yield that is significant is still a challenge. Why don't we consider a different stream, which is hydrolyzing it using existing platform ESIT for the characterization and processing, while considering supporting it as a cultivated meat medium. So this is how I come to this field with zero background coming to cultivated meat. So it was fortunate that James Cook University, together with SIT, has able to identify key collaborators, including, the listed IPB University in Indonesia, because seaweed is available and is cultivatable within the Asian countries. Of course, in other regions as well. So we are in collaboration with them to add more data source or the raw ingredient as a screening starting material. In addition, we are working closely with Umami Biowork for the screening and the testing for cell culture. So the processing and evaluation is done together by SIT, as well as James Cook University. Well, the end point is to make sure that this ingredient and media after the process is considered scalable and cost-effective, passing and suitable for the cell culture testing. Of course, the safety aspect is quite important as well. With this project, it has helped me to expand my capability coming to research, because in the past I was all doing the mostly plant-based or other alternative protein sources. But consider using the same material and expand it to a different angle or more value-adding application is something this program has helped me. In addition, in the past, although I have come across with some of the meat cultivation or cultivating meat like a related industry or startup, but working closely with them to understand their problem and also the safety attributes and what is the processing or the property that is required in the cultivating media is still something that is adding, or say, helping me to build my expertise in this field. I won't say my domain expertise is in this field, but it was lucky that I have this network that helped me and also help this project to go much further. So I agree with the previous I echo what she said has mentioned that it is quite important to find key collaborator that has complementary expertise, just like what Dr. Deepti, right? Lily, did I mention her name correctly? Okay, thank you. Forgive me if I say it wrongly. So as what you see that this project really start from, just a lab visit. Okay, to understand each other. And more important is how to make sure this is a tangible solution that is scalable later to meet the industry need. That is only a starting point, and we have to sit down to meet the industry to understand their pain point, and therefore to develop solution that is suitable for their current situation. And the application side, actually, I don't have much to share except that I echo with what Dr. Deepti has mentioned. It's quite simple and straightforward. It's easy, and if you want to learn about how to do it, actually you can just go online and do a mock submission. It's not taking too much time. And coming to the submission, I would say, yeah, it just took us very short time to figure out which part is fitting into what. But later the submission, after everything is done, including, all the files such as budgeting, the proposal, it's pretty straightforward. My only advice is make it pretty lean and information that is more direct, targeted to what you are trying to do and trying to deliver. And I was very fortunate to get selected and shortlisted for this awarding. And it is a great achievement, I would say, because that helped me to expand my research to something else, forcing me to learn new things. That is the most exciting part, I would say, about this project. However, coming to delivering the project, I cannot do it on my own for sure, which is another skill set to make sure that, okay, to deliver it, we have to work closely to understand each other's research field. So the communication is quite key in this project, as what you can see that my expertise only coming to the processing, evaluation, and also amino acid screening. So that is most of the sharing. And how does it help me? I would say career-wise, it has built me a further milestone to expand to a different field. I won't say I'm building my expertise in that field, but I feel more, self-achieving, coming to, do something more and expanding my knowledge to understand, okay, actually seaweed protein can do something else rather than what we have been doing in the past, okay? So that's most of the story about here. And as what I mentioned, lab visit is start of the, all the story. So you are very welcome to visit us in Singapore. And our lab is open all the time to the public and of course to the researchers as well. So we are open to, student to visit, scholar to visit us, as well as hosting you for some other event like seminar. You can come to us to a short trip if you pass by Singapore for 4 hours. Yeah, take a taxi to come to SIT, stop by for 30 minutes and have a coffee and go back to the Changi airport. It's not a problem. Okay, that's what I can share, as well as A-star CP. Yeah, I would say maybe together. Yeah. Okay, that's most of my sharing. Yeah. And in the beginning, I was a little bit hesitating because this is not my domain knowledge coming to cultivating meat. But the key is to find a partner who has the expertise coming to proteomic profiling and meat cultivation and all these kind of things. So the trust is building up, and when we come together and find the niche point, it's quite interesting, everyone who doesn't know each other, and finally we find out a sweet spot to work closely together. Okay, that's most of my sharing, and thank you for your time and attention.
Yes, thank you very much, Prof. Yuan, for that wonderful presentation. And I really like the point you made, and I believe Dr. Deepti mentioned this too. It's basically about being new to the alt protein field, but not letting that be something to stop you from actually seeing that as an opportunity. Because one of the big points we have at GFI is that where the alt protein industry is right now, we actually really need minds like yourself who are experts in characterization, metabolics, things like that to come in, because the problems that we're facing, the bottlenecks that we're facing in the alt protein industry are not general food science problems, right? We need people with the characterization expertise, with all of these expertise from different fields to come and apply. And I think to your second point about to make a really successful and thorough application, you really do need people with other strengths to come in, right? So I think you making that point really clear about where you fit in this picture, but where your collaborators are coming in to strengthen. And this point was mentioned by Dr. Deepti as well. So thank you for reiterating that and showcasing that again. And yeah, that the collaboration is really important. So yeah, that was really cool. And I think for me, really the big point is whether or not you have experience in alt proteins, this is really just a great chance to get in and explore and expand that skill set and get that excitement about science, right? So yeah, thank you so much for sharing. Any last thoughts now before the Q&A, or all good? I think all okay. So with that, yes, thank you again to sorry, to Prof. Juan Du for sharing your experiences. Now I'd like to pass on to Dr. Nazneen Sofio from A-Star.
Hello, good evening, everyone. So give me a moment to share my slides. So I'm just gonna not talk much about the research part, and can you see my slides?
Yes.
So I would start off with a small introduction, though Aaron did a extensive one already. So I did just to give you an idea of my background, so you can see I did my bachelor's in biotech, did my PhD in biochemistry. I'm a senior scientist at Singapore Institute of Food and Biotech Innovation, but I work mostly on metabolic engineering, synthetic biology. So you can see it's not very like all protein focused, but I can talk about how I bring that background into all protein research. So what I work on is working on sustainable biotechnology to produce microbial lipids for varied applications. So, one important ingredient for alternative protein is alternative lipids, because the proteins needs to be tasty even, right? So the lipid gives it the flavor, the texture sometimes. So it's also important to look into these other components of all proteins, and bringing my biotech background into this all protein scene was more like making microbial lipid using synthetic biology, and make different kinds of, probably these all lipids can not only go into alternative proteins, they are also it could be used as vegan labels and go into infant formulas, supplements, and different other applications. So that's what my main research focus is. I will quickly move on to my journey as a GFI grantee. So I did my PhD in 2019, so not so long ago, and started my postdoctoral research sometime end of 2019. And this opportunity to apply for a GFI research grant came up mid 2021. So it was like I was quite early in my postdoctoral journey. So you can really call me like a very early career scientist at that point. And it was an international grant, and so I was a bit hesitant. But I had like a team of collaborators and a team which I became a part of. And then when I applied for this grant, actually, we looked at the RFP, and we looked at different specific problems that were stated in the RFP call. And then we figured, then we had an idea already in place, which could very nicely target one of the specific problems. And then we applied for it, and we got the project sometime around 2022, and which ended in 2024. So as I said, GFI gave me a very nice platform to kind of step on and advance my career as an early career scientist. Actually, this was the first grant I applied at all. I haven't applied for a grant before that. So getting that success was also a boost. So mostly I would like to say that even if you're early career, just don't let that hold you back. If you have novel ideas, if you have interesting ideas, just go ahead and pursue them. It's very important. And GFI was very kind to also do a blogspot, which gave a platform, which gave us a platform to showcase your research on a very international platform. So that also helps the grantees. So GFI not only gives you the fund, it really supports your journey, initial journey, to give you a platform to make yourself visible, make your research relevant, and to even connect with all these other experts in the field. So as of now, for example, I still have contacts of some of the grantees that were granted at the same time as me. So I can reach out to them, and that kind of gives me a more base of collaborators I can reach out to. So it's good for networking. It also gives you a very, very good platform to network with collaborators around the world. So yeah, if you see someone having an expertise that is complementary to you, you can reach out and write a grant together. Yeah, I mean, since it's international grant, it doesn't matter where you are. I mean, the funds can get sent to different partners. So yeah. So it was, as I already mentioned, it was my first grant as a lead PI, which I got, and also gave me my first corresponding author paper. We published the research that we did as part of the project already sometime last year. And not only that, we generated a lot of data, so I could go on publishing it for probably a few more publications, and gave me a lot of data to kind of go for other grant calls that I can use from this. So it was really like a launch pad, and I would really encourage early career people to pursue this, as has been said already by previous two speakers, that the application process is really simple. So you don't really need to worry too much about, am I writing it well, so it's quite simple. So it gives you that idea of how to go ahead and write a grant. So if I have to share my grant writing insights specifically from this project that I wrote, first and foremost thing I think that really helped us was that we addressed a very specific problem statement in the grant call. So our idea fitted into that problem statement very nicely. I think it was also mentioned by Deepti in her slide. So read the RFP and read the focus areas, sub areas, and figure out like how to tailor your proposal to that specific problem statement. Also, as I think already has been talked before, extensive collaboration. So as I said, we had different collaborators itself at CIFB, where so at A-Star basically, and we were having a different expertise. Some were strain engineers, some were doing fermentation, others were doing analytics. So it's very important to get the right team together, and the grant proposal should also be like a solid teamwork, not just the project itself. So the grant proposal needs to like highlight what all the different collaborators are doing and what novelty specifically, what novelty are you bringing in? I think that's, if you can highlight that very nicely, then it's a win-win situation. Apart from that, I think this part has also been touched upon about relying on your back expertise, even though like you are just a part of the whole puzzle. You are not just the one doing everything, right? If you have a collaborative team, you're just part of the puzzle, and then rely on the expertise and how you can contribute to the problem statement or to the idea. So as you can see, I'm not doing any cell cultures. I'm not doing any alt protein work before this, but I'm mostly working on alternative lipids, but I'm still relevant because protein is just not the protein. There's a lot of different components that goes into there. So you can look for that and how you can then contribute, I think, yeah. And if you have some preliminary data from your own expertise or background, then yeah, just add it onto the grant call, so it kind of boosts your proposal. Yeah, I think that's all the slides from me. And also thank you to GFI for many opportunities and many platforms that has been given to me since 2021. So yeah, really grateful for that. Thank you.
Thank you very much, Dr. Sophia. I think it was really like the points you highlighted at the end with that chart was very, very cool to see about like, yeah, I didn't even think about the preliminary data until you actually brought it up. I think that was a really cool and interesting point that you got to showcase. And then, yeah, again, on the collaboration point, and yeah, I mean, always so like cool for me to remember that this was like the first grant you got. I think that was so exciting, and getting to work on a really impactful project. So yeah, and I think also on the APAC team, like Singapore is one of the countries that we cover. So like Dr. Nazneen Sukio and Prof. Dujuan is such a important part of our community as well. So like really, really great to have them both. And I think with the idea of all protein dialogues, like getting to interact more with Dr. Deepti and really bridge our communities together. I know Dr. Deepti plays a really big part in the GFI India community. So I think with this whole platform, having all three of you come and share your insights, seeing like the similarities and differences, and also on my part, getting to know all of you more, I think is really great. So thank you again to all three of you for your sharing. I do want to jump to some Q&A. We do have 15 minutes left on our call. So I think anybody in the audience who does have any more questions, please feel free to put them in. I'll ask the questions in our Q&A chat right now. We have two. I also have a couple questions myself to maybe ask and add on, but yeah, obviously we don't have to use the full 15 minutes. So let's see what the audience has, and then we take it from there. So our first question is maybe actually if I'll ask a question, and whoever wants to answer first, please go ahead. And if everybody wants to contribute a little bit, yeah, just go in and add stuff. But thank you for your interesting presentation for the application. regarding the impact of your research and potential to the industry, can you give us an advice for the application? So I think this is questions about the application process and then including the impact part in the application. I believe this question was asked during Dr. Deepti's talk, so maybe I'll start with her. Like, how did you include the impact element into your research question? And maybe how did you frame all of that being new to all proteins? Like, how did you find that data, information, things like that?
Yes. So, yeah, thank you for your question. So, so it's when I say impact, the impact is not just the societal impact, but the impact on the industry as well. So, when we are actually working on solutions, we try to understand how will our solution help in future in overall shaping the research in the alt protein domain. So we focused on two aspects, societal aspect as well as commercialization aspect. And we tried framing it in perspective that while it is just not the research which is limited to the laboratory, but it is making a lot of impact on everyday life of person, as well as it's making a lot of impact on commercialization. So I think in my knowledge or in our understanding, I think we as a team were focusing more on any problem or any solution making a social impact on science, the science making a social impact. So I think we kind of added it in the introductory section itself. So, and we also concluded while adding the impact on the commercial side. So this is how we framed it. But yeah, I mean, anybody else might have a better perspective towards it. So yeah.
So maybe I'll pass to Prof. Chuandu.
Sure. In our project, actually, we do have a professor and also who has the domain expertise in techno-economic analysis. So in this perspective, meaning that all the step of processing or proposed solution shall consider, taking the technical perspective together with the economic analysis together to make sure this is a scalable and cost-effective solution. So that expertise, I think, although Professor Paul Charlotte is not a full scientist who has the major domain knowledge in chemical engineering, but the principles and the philosophy behind it is the same thing. Is when we are generating a solution for processing a product, we have to keep this in mind. Is this addressing the issue in the industry, and how is the cost? Of course, there are other attributes such as the sustainability attribute. The life cycle assessment will be implemented, but without cost, such solution will not run for long term. Okay, so taking on someone who does not have your expertise to support this part of the analysis, while making a true impact for the community and also industry will be quite helpful.
Thank you very much for that answer, and I'll pass to Dr. Sophia.
So I think mostly it was covered by both of them, but I would like to add that for the impact and the potential in the industry, sometimes you just don't know if it's a very, very like low TRL idea. It's like very new idea. So I think what happened to our project was when we started off, we just tried to make a product that could be potentially interesting. But after we achieved that, and then we went ahead and presented at different conferences, then where these industry members were present, they would approach us and try to have a conversation. So get into like, so basically you just like need to have the idea that this is the product that might be interesting, and you try to make something like that. So yeah, so that can also help if there's not many industry partners in your grant call. Yeah.
Wonderful. Thank you so much. So I think adding on to Dr. Sophia's point and just maybe summing up from the other speakers as well seems like, yeah, things like techno-economic analysis, like social impact is another one. And then yeah, the partnership space part coming in. And then I would also just maybe share that on the GFI website too, like we do have like quite a few reports on the impact of like alt protein technology, things like that. So like for inspiration and understanding like what solving certain bottlenecks in the value chain for like cultivated meat or for fermentation, for example, can do. You can also go to our website and try to like maybe reference or like learn from those as well to like inspire what you put in as the impact. Right. But yeah, thank you so much for those questions. We did have a few questions about like startups, but I do believe some of those questions are addressed in the FAQ, but maybe I want to ask like, were there like, if you all could share about like any industry or like any partnerships you guys made and how you went about doing that. I know Prof. Dujuan, you specifically had a list of some of like the industry-based ones. If you can share about how you did that, that would be great.
Sure. In our project, we do have industry partners. And in general, basically our school, SIT, is aimed for applied learning and applied research. So we do have this like a pool of industry available to approach. But more important is some of the workshop that have been hosted. For example, the Good Food Institute, like some of the conference and workshop, is a great opportunity to learn about the industry and other parts. I would say there could be other events, related to food, such as conferences and others, that you can go and approach the company to understand what is their need. That's basically the ways. In addition, as a university, we always send students to their intern companies. Sometimes the company would like to invite us, the mentor or supervisor from the school to visit them. So there are one way or the other to build connection. Usually because in Singapore, we are a small city state, approaching and understanding each other's need is quite convenient. Yeah. So that is the situation here. But I understand in other countries, yeah, there could be some of the local or national conferences that you can go and approach them. Yeah.
Dr. Deepti or Dr. Sophia, do you have anything to add?
Yeah, I think I can just add on to Prof. Chuandu's comment because I'm also from Singapore. But all the institutes, also A-STAR, we do have like a dedicated team or a dedicated pool of industry that we engage with. So if your institute has something like that, it will be good to reach out to them and share your idea with them and say if they can connect you with someone in the industry. Like that's what I generally do, not just for this grant or any grant. If I have an idea and I want some industry partner to like listen to me and tell me if they really want to support my grant, I would just go to we call it the business development team, or at least so you just go to them and just ask them if generally they would have contact with different industry partners and they can connect you with them. And yeah, so just know how it works in your specific institute. Yeah.
Yes, I think I completely agree with Dr. Sophia. In India, we also have institutes where there's a department which is dedicated in finding partners to support you, to collaborate with you. So we also have an ecosystem where there are a lot of startups. Something like IIT Bombay is one of the example of such ecosystem where you find academics, you find a lot of industry support, a lot of business development activities keep on ongoing. However, I still feel that in being a vast geographical area, these efforts are still concentrated in some cities. We may or may not have a complete directory of neither academic labs, research labs who are doing research in vast areas or different fields, neither the industries who are locally doing some amazing result. I really don't think so that we kind of have a very common repository, a structured repository in that sense in India. And I think it would be great that if we have one soon, at least in the food technology or food related domain. But yeah, so in most of the cities now, we have the platform in the major cities in India, at least.
Wonderful. Thank you so much for all of those detailed insights. I think this was especially maybe helpful for the last question we have in the Q&A chat right now about early stage where the concept is promising but needs refinement, industry mentorship, or collaborative validation. So I think all of these networks are which were mentioned, how to reach out. And then at GFI, we are like ecosystem builders and connectors. So I think all of the different affiliates will have like support on like trying whatever we can do, either resources or support on connecting with people. We also have airtables of like contacts and stuff on our websites as well. So yeah, definitely feel free to like explore on the sites themselves for your affiliate region too. Yes, thank you so much. And then maybe one a little bit of a technical question for Dr. Sofio. Do you just work on microbial lipids alone, or do you work on microbial proteins as well?
So as of now, my focus is mostly on microbial lipids, but in the past, I've worked with different kind of fungal proteins too. So I do have the expertise to work on it, but recently all my projects has been mostly focused on microbial lipids. But yeah, I do have the expertise and the experience. Yeah, feel free to reach out if you want to chat about something. Yeah.
I think to that point too, like, and all three of you kind of mentioned this during each of your individual parts, is like, for the grant call, I think it very explicitly states out like what the grant call is looking for and what the grant call isn't looking for. I think Dr. Sufio especially made this point in the image at the end. So I think it's very clear exactly what the statement is. And yeah, I believe Dr. Dipti and Dr. Joandu also mentioned that like the keeping it things like lean and clear. And I think all three can attest to the fact that like scientific rigor is like the top priority for the proposals that we're looking at. So yeah, just the question about like lipids versus protein made me think too that like make sure everybody really pays a lot of attention to like what's on what they're looking for, what they're not looking for, and especially read the what they're not looking for part, because I think that helps clears up a lot of questions. Cool. And we do have a few more questions, but I will recommend the attendees for in terms of like the requirements needed for the application, to make sure you check the FAQ doc, because it does quite explicitly state that. And if you do have any more questions, there's also like an email. If you have like a special context or situation, you can email the RGP. There's an email there where it goes through like the global organization, where they can specifically answer things about your like eligibility for the program. I want to leave the last like 2 minutes for any final thoughts, maybe just like a thank you, good luck. I know you guys already said a lot, but maybe like a sentence or two from our speakers before we close off. I'll start with Dr. Deepti, and then we go to Prof. Juan and then Dr. Sophie.
Yes, I think the statement has been I think by all the speakers today, and it's been spoken out so much. But I think find correct collaborators and focus on the problem statement. And I think that's what is more important to make even the impact. You need funding. So for getting the funding, you need to have the correct proposal in hand. And for getting that correct proposal, I think finding a correct team and addressing the correct problem. So ultimately, I think only then you will be able to make the impact. So I think that's what it means for us. So thank you, GFI team. Yeah.
Wonderful. And Prof. Dishwan?
Sure. My suggestion is go a little bit wider and also, encourage yourself to reach to people who might not know that well. And you never know what is the response and attitude. If you are able to work with someone who is not in your field at all, it doesn't mean there will be difficulty communicating with each other. You might find something more exciting. Of course, the scientific approach the rationale has to be valid and strong. But for you to do that, you need some other people who has a different domain knowledge. Okay, so again, that is echoing what Dr. Dipti is mentioning. Find the right complementary partner.
Wonderful. Thank you so much. And Dr. Sophia?
I think most of it has already been spoken. So I would just say that if you believe in your idea, that you can really make an impact with it and just highlight its novelty, go for it. make sure your scientific base is solid, and that yeah, you can so that's the way you can really convince the reviewers. Yeah. Okay.
Wonderful. Yeah, thank you so much. I think it's great to like just take these key points home. So like, thank you so much for just verbalizing that one more time for all of us. And yeah, I think with that, I want to conclude the part 2 of the 2025 Research Grant Program webinar series. And with it, our first installment of Alt Protein Dialogues. And I think I'm just really excited because I mean, all 3 of you are such big champions to our Alt Protein community. And I'm really happy that you know, you guys are like our entire, like our bigger community now from APAC, India, GFIC, Australia, New Zealand is all getting like visibility to your great work. And you guys are all connected more so as well. So huge thank you for our speakers for sharing and everything that you guys do, the great research, for joining us today. We hope that this session has provided valuable guidance on structuring a compelling research proposal for the GFI RGP. All the best to everybody here. We hope to get proposals from all of you. I hope you guys are very inspired. And we'll be sending up a follow-up email with a recording of today's session, along with relevant links and additional resources. If you have any further questions, please feel free to reach out to our team. And yeah, please feel free, make sure to read the FAQ document well as well, because a lot of the questions are answered there. Thank you again. We look forward to seeing your application. So, and yeah, stay tuned for more Alt Protein Dialogues webinars. We'll be addressing some big questions, which will be relevant for our large community as well. So yeah, with that, I will close off our session today, and thank you again to our speakers.
Thank you.